Cell-mediated immunity is the form of immunity carried out by the T lymphocytes, or T cells. The body contains millions of different T cells. Each contains a unique form of T cell receptor on its membrane. Like immunoglobulins, T cell receptors are produced through a process of genetic recombination, which is able to produce millions of variations. Each T cell receptor is capable of binding to a specific foreign antigen, but only after the antigen has been processed and presented by some other cell. T lymphocytes recognize such presenting cells by means of another surface protein called CD. There are two types of T cells, those with CD4 on their surface and those with CD8. CD4 receptors are found on helper T cells, while CD8 receptors are found on cytotoxic T cells. CD4 and CD8 T cells are activated differently. They also play different roles in the immune system. Most cells that display CD4 become helper T cells. CD4 T cells can only recognize foreign antigens when they are presented in conjunction with major histocompatibility complex, or MHC, class II molecules. These protein molecules are only found on certain immune system cells, called antigen-presenting cells. The helper T cell antigen receptor must match the presented MHC2 antigen. Their binding with one another is the first signal needed to begin activation of the T cell. A second signal, known as co-stimulation, must also occur. Pairs of molecules attached to the membranes of the T cell and the APC cell help bind the cells tightly together and can provide co-stimulation. Cytokines, small protein hormones, are released by the antigen-presenting cell and are co-stimulators. Once the T cell is activated, it proliferates into clones of itself. Most of these new cells differentiate into effector T cells. The receptors on these cells are able to bind to the same antigen as the receptor on the original T cell. Some of the T cell offspring will not become effector cells, but remain as memory T cells, which will respond rapidly upon future exposure to the same antigen. Once activated, helper T cells secrete cytokines. One cytokine is interleukin-2, which is important in most immune responses. It is also known as T cell growth factor, since it acts as a co-stimulator for T cells. It also enhances the activation of B cells and natural killer cells. Other cytokines are interleukin-4, or B-cell stimulating factor, and interleukin-5, which is also involved in B-cell co-stimulation. In addition, helper T cells can secrete gamma interferon, which stimulates phagocytosis and nonspecific resistance. Gamma interferon is involved in both cell-mediated and antibody-mediated immunity. In general, individual helper T cells do not secrete all cytokines. Subsets of helper T cells secrete different cytokines. The subset of helper T cell that is activated is determined by which defense is needed against a specific pathogen and will also determine which cytokine is secreted. Most cells that display CD8 become cytotoxic T cells, also known as killer T cells. CD8 T cells recognize foreign antigens when they are presented in conjunction with major histocompatibility complex class I molecules. Virtually all nucleated cells in the body, including antigen-presenting cells and lymphocytes, express MHC class I molecules. T cells that recognize self-peptides expressed by class I major histocompatibility protein on the surface of healthy cells don't survive in the thymus gland. Surviving T cells recognize certain tumor cells that express abnormal proteins seen as foreign, cells of a tissue transplant that express foreign class I MHC, and body cells infected by viruses which have viral antigen on their surface. Binding to an antigen presented by a foreign tumor or viral infected cell is not enough for activation of cytotoxic T cells. Co-stimulation, provided by the close association of molecules on the two-cell membranes and by interleukin-2 and other cytokines produced by helper T cells, is needed. Once the cytotoxic cell is activated, it will proliferate into T cell clones, each expressing the receptor of the original cell. Most of these clones differentiate into effector T cells. The receptors on these cells are able to bind to the same antigen as the receptor on the original T cell. Some of the T cell offspring will not become effector cells, but remain as memory T cells, which will respond rapidly upon future exposure to the same antigen. Cytotoxic T cells can eliminate cells that are infected or have become cancerous. They also act against foreign cells in two ways. The first mechanism is perforin-induced cytolysis. Perforin, released by cytotoxic T cells, punctures holes in the target cell membrane, causing the target cell to burst. The second mechanism is lymphotoxin-induced cell death. Lymphotoxin, also secreted by cytotoxic T cells, starts a cascade of events in the target cell leading to degradation of its DNA and cell death.